Hi, I'm Sachin Velamkunda, and I'm a research assistant within the Division of General and Internal Medicine at Brigham and Women's Hospital. Today, I am discussing identifying and reconciling patients' allergy information within the electronic health record. First, here is background on allergy information in the EHR. The allergy list in the EHR provides important information to clinicians that is used to ensure patient safety and trigger clinical decision support alerts. Issues arise when the information contained in the allergy list is inaccurate and incomplete. Insufficient information about allergies has been attributed as the cost for up to 12% of medication errors. The information is often split between different sections in the EHR and documented as uncoded free text outside of coded allergen and reaction fields. Allergy deletion is infrequent in the EHR, and this leads to unnecessary drug allergy alerts that contributes to alert fatigue and physician burnout. Given all of this, we set out to develop methods for systematic allergy reconciliation within the EHR to reconcile allergy information from multiple sources, as no tools exist in the EHR to date. This study was a retrospective cohort study approved by the Mass General Brigham IRB. MGB is a nonprofit integrated healthcare delivery network located in Boston, Massachusetts. In the study, we included all patients who received care during 2018 at any MGB site. We collected demographic information, allergy list, laboratory data, drug allergy alerts, oral medication challenges, medication orders, coded reactions, and free text comments. Using this information, we developed methods to identify discrepancies between their allergy list and collected allergy information. The aim of the study was to examine allergy documentation within various EHR sections and to better understand the prevalence of incomplete and inaccurate and redundant allergy information documentation and identify approaches for extracting and reconciling allergy information in the EHR. Here are our methods proposed for a systematic mechanism to identify, compare, and examine allergy documentation in 4 EHR sections and their discrepancies. For free text comments in the allergy module, we calculated the percentage of allergies that had reactions mentioned in the free text comments field. We further investigated whether these reactions extracted from the free text were available as coded options in the AHR or as reaction concepts in our value set. To do this, we ran the text through our natural language processing system, mTerms, to identify reactions and compared those to coded reactions. The reference allergen table in the EHR contains over 42,000 allergies, among which many are symptoms, duplicates, brand names, or other variations. We found that it is possible to enter duplicate allergens to the same medication in our AHR. For example, a patient could have piperacillin, tazobactam, or zosyn as 2 allergens on their list. We developed mappings in the allergen database and identified preferred allergens that a duplicate would be mapped to. Using this mapping, we were able to identify patients with duplicate allergens and would be able to consolidate them into a single entry. Using the laboratory results, we were able to identify patients with a positive latex IgE result. We pulled their latest result value and compared the results with the allergy list to determine patients who required reconciliation. For these patients that we identified who did not have an active latex allergy, we would suggest an addition of this allergy to their medical record. For oral medication challenges, we chose to focus on penicillin allergy challenges. We identified patients who had an allergy to a penicillin removed and at a later date had an allergy to penicillin re-added. We then reviewed patients' charts to determine if the re-addition of the penicillin was valid based on if delayed reactions were reported or if the patient had reactions to subsequent penicillin medication after passing the challenge test. For discontinued medications, we focused on sulfa drugs and ACE inhibitors. Using the medication order history, we were able to identify patients who had a medication order discontinued with the reason being allergic response, but did not have the medication on their allergy list. For these patients, we reviewed their charts and suggest that the allergy be re-added to their chart. Over 1.5 million patients were included in the study, of which 45% had an active allergy, resulting in a total of over 1,751,000 active allergy entries. The majority of our patients in our cohort were white and female. Delving into our findings, we see approximately 50% of allergy entries had missing reaction information in the allergy module. Of all the active allergies, we found that 20% do not have a coded reaction, and 26% contain additional free text reaction information that can either be coded using our existing reaction lexicon or potentially be coded with an enhanced reaction lexicon. First, delving deeper into free text comment entries, we see that overall 43% of allergy entries had a free text comment, and 25% of all allergies had at least one reaction mentioned in the free text. 12% had a coded reaction, and 14% had a reaction from our value set. These are areas for reconciliation. The table shown here depicts the most frequent reactions found in free text, including rash, vomiting, hives, and GI upset. Regarding duplicate allergen entries, among the patients with allergy entries, we found that 3% of patients with a total of 53,000 duplicate allergens on their allergen list. Among these patients, 10% had 9,000 orders with a medication that triggered a DAI alert for their duplicate allergens, and this resulted in an additional 10,000 drug allergy alerts. Reconciliation of allergens would remove almost 27,000 allergens from patients' allergy lists and would have prevented 10,000 duplicate drug allergy alerts during the study period. The table below showcases the top 10 most frequently occurring duplicate allergens. In regards to medication allergy laboratory test results, where we focus on latex IgE, we identified 16 patients without a documented latex allergy who had a positive test result, of which 12 had a positive test result with high severity. In regards to oral medication allergy test results, where we focused on penicillin, in our cohort, 737 patients had a documented penicillin challenge order. After reviewing the results, a total of 55 patients had a penicillin allergy removed and subsequently re-added, following the challenge test. After thorough review, we found that 36 of these patients had the allergy inappropriately re-added. Finally, in regards to medication orders discontinued due to ADRs, there were a total of 1,366 ACE inhibitor and 969 sulfa antibiotic orders that were discontinued with the discontinuation reason allergic response. We manually reviewed charts for 137 ACE inhibitor and 174 sulfa antibiotic randomly selected orders from unique patient charts and found that 34% of the reviewed ACE inhibitor orders and 17% of the reviewed sulfa antibiotic orders needed reconciliation, as the allergies had not been added to the patient's allergy lists. Our main finding was that 266,275 patients' allergy records indicated the need for reconciliation based on our methods. Reconciling reaction information from the allergy module's comment section into coded fields is important for downstream CDS. Based on the discrepancies identified, we proposed several solutions that, when combined with the allergy regulation tool, may improve the quality of allergy documentation in the EHR. One potential solution involves the enhancement of the predefined list of coded allergens and reactions. Synonyms of reactions such as edema for the coded reaction swelling are not searchable terms, and this may lead to clinicians entering these more granular reactions as free text. Expanding our coded reaction list and synonym lexicon is an important solution for this issue. In addition, modifications to the EHR design could potentially make updating the allergy module more compatible with efficient workflow practices. Furthermore, there were several relevant limitations to our approach. Our discussed algorithms were developed at one large healthcare system located in the northeastern United States operating a commercial EHR. Likewise, more extensive examination of different test result types and including more data may be further valuable. In conclusion, our study suggests that incomplete and inaccurate allergy lists are common, with 37% of patients with allergies having a discrepancy using our methods. We also found that our methods can be used to both identify allergy information in different areas in the medical record and be used to reconcile patient allergy lists. Our findings further highlight the need for automated reconciliation methods within the EHR. Providing clinicians with the tool to improve the accuracy of allergy lists is important, as it ensures patient safety, improves clinical decision support, and provides more relevant alerts. Lastly, I would like to also take a minute to thank both my research team and my mentors. Thank you for your time.